acids are amazing. I mean, who wouldn't love them? Well, maybe people who don't know enough about them. So let's talk about amino acids. All of them. Let's go.
So I'll get into some technicalities in a minute about like what actually defines an amino acid and how amino acids can be more than just protein letters. But typically we think about amino acids as these building blocks of proteins or these protein letters. They have this generic backbone, which has this amino group and this carboxyl group that we'll get into that give them their name and also provide them a way to link together into chains using that generic part. They link up through these peptide bonds. And then they also have, when they form these chains, so now you have a polypeptide, it'll fold up based on this unique part, this R group. So each of these amino acids has a generic part as well as this unique side chain or R group. These R groups, as you can see, have this very diverse properties. Some are big, some are small, some water likes to hang out with. So these hydrophilic ones are often found on the surface of a protein. Some water doesn't like to hang out with. These like hydrophobic ones, these non-polar ones are often found on the inside of a protein. So basically what happens is that these get linked together in this process called translation based on instructions from messenger RNAs. And using these ribosomes, which is this big complex of proteins and RNA, that's going to read out the instructions and have the corresponding amino acids brought to them by these transfer RNA molecules, these tRNAs. And then this process called translation, the amino acids that are coded for in this messenger RNA. So the messenger RNA that's a copy of the gene with the original instructions. Now, the messenger RNA carries a copy of those instructions. The tRNA brings the corresponding amino acid, and in this way, you can link up amino acids, distinct orders of amino acids, into these polypeptides. And once they're linked together in this translation, then they start coming out of the ribosome, they start folding. And how they fold up is going to depend on the properties of those amino acids. So the properties of those side chains, how those side chains can interact with things, as well as how the side chain actually can limit the movement of the backbone. And so we'll get much more into all of these different things. But the amino acid itself is going to be what we call the sequence of amino acids is what we call the primary structure. And it's of primary importance to how the protein folds and functions. And once these amino acids link together, we call like the leftovers residues. So what was this R1 this is now a residue, or this is a residue. And so we can call those residues. Often we just call them amino acids, even still when they're in the chain, when we're talking about like a specific amino acid position, it's technically a residue at that point. So you know that term. We'll get much more into how the protein folds and functions, as well as more about the different proteinogenic amino acids, so the ones that we get in proteins. But first, let's just clarify some terminology with regards to amino acids and how there are some other amino acids that we typically don't think about very much and that don't come up that much in order to be fair. There are 20 common genetically encoded ones. We'll get more into what that means. There are a couple of other ones that can sometimes sneak in there. So we use selenocysteine in our bodies. Pyrrolysine is only used by some like bacteria and archaea and things, and more on these later. But these are our 20 main ones. We commonly think of them as being like amino acid letters, being like protein letters. So these are the building blocks of proteins, and they link up into these chains, which will fold up into proteins. And as we'll see, the different properties of this amino acids are going to influence how those proteins fold and thus how they function. But amino acids aren't just these protein building blocks. They also have other purposes. So for example, they can be used as hormones. They can be used as signaling messengers. And there are also things that are technically amino acids, but we don't typically think of them as amino acids, like creatine, which is a molecule that can be used for energy storage. in the form of creatine phosphate, and it's actually made up of amino acids, but it still has an amino group and a carboxylic acid group, and so it is still technically an amino acid, but these are the main amino acids that we talk about, and we can characterize these as alpha amino acids because everything is kind of happening on this central alpha carbon. So here, this alpha carbon you have attached to the side group, so this R, the carboxyl group, and the amino group. Speaking of this carboxyl amino group, so this is where we get the name amino acid. But when we typically see an amino acid, it's actually in its zwitterionic form, which means that the carboxylic acid group is actually in its deprotonated carboxylate state, and the amino group is in its protonated state. You often see it drawn in this like unrealistic non-ionic form. So an ion is something that's charged. And so typically we have this zwitterionic form, like this is that like actual form that we see most of the time in our bodies in solution, that sort of thing. And in this form, it's neutral overall, but you have this positive part and a negative part. And so we call molecules like this, where you have a fully positive part and a fully negative part, as opposed to just partial charges like you might see with the polar molecule. We call these zwitterionic, which is a really cool name. But anyways, this is the form that it like more realistic form, but it's often drawn like this, emphasizing the carboxylic acid, which is where we get the name amino acid. But when we see it, it's typically already acted as an acid, meaning that it's given up a proton, so it's given up an H+, and now it's in this conjugate base form, this carboxylate form. And we'll get more into acid and base chemistry later on when we talk about some of the quote unquote acidic and basic side groups. But this is just something to keep note of. So basically, if you go farther down, so you go more acidic, farther lower in pH, more acidic, more protons, this is going to be protonated. So you would have a carboxylic acid group, but then this group would also be protonated. So you'd end up with a +1 charge. And if you go more basic, so you go to a higher pH where now you don't have enough protons available, this is going to deprotonate. But so this is also going to be deprotonated. And so therefore, this is going to be negatively charged, and you're never going to have something that's actually like this. Instead, you're going to have something like this. And we'll get more into this sort of things later, but just so you don't get confused, that's why you might see it drawn like this, but typically it really is like this, as well as why the terms like carboxylic acid and carboxylate, it's because they can go back and forth between those two states. But because this carboxylate group is so happy like this, you're typically going to find it in this deprotonated state as opposed to in this carboxylic acid form. But in the context of a protein, you're really only going to see these things come up at the very ends, because as we'll see, these proteins link up using those amino groups and those carboxylic acid groups. So as I mentioned, there are these 20 common ones, and then I said there were a couple of weird exceptions. And we're going to talk mostly about the common ones and how we can classify them based on their different properties. But first, I just wanted to mention these couple other ones, and I have whole posts on these if you're interested. But so selenocysteine, so this is cysteine. This is one of our like common ones, and so this goes into proteins and it has the sulfur group. Selenocysteine is a version of that has selenate instead, that has selenium instead of sulfur, and so it has slightly different properties. So in the case of selenocysteine, what happens is that in the messenger RNA, so in the instructions for making the protein, there's downstream, there's this RNA element. There's this sequence in the RNA that makes it fold up into this shape that then binds to this protein, which then binds and helps bring in the amino acid selenocysteine, it's RNA carrying selenocysteine, to be inserted instead of the stop codon. So it's this pretty cool stuff, but you're bringing in this selenocysteine, so this tRNA, based on a sequence downstream of what would normally be considered a stop codon. So for a codon that instead of having saying, okay, put stop the protein stun, this codon would normally specify that this codon normally specifying that that's the end of the protein, and then a release factor would come and bind and cut it off. Instead, in the case of selenocysteine, you have this selenocysteine charged tRNA hanging out right nearby, attached to this RNA with the help of this protein, and it can kind of sneak in. So we use selenocysteine in some proteins in our body, a few of them, and they're really important ones involving like redox things, so reduction and oxidation, which I'm not going to get into. But selenocysteine is really good for sensing things like that. And the reason, so we have it in our proteins, not many of our proteins, but we do have it in our proteins. So it is like a proteinogenic amino acid, but we don't typically think of it as one of the main 20 because it's not like genetically encoded in the traditional sense. It doesn't have like its own codon, and it actually doesn't have it, like it gets modified. It does, the tRNA for it doesn't get charged with it. What I mean is that in amino acid, when tRNA charging, that the amino acid, when it gets added to the tRNA that has the corresponding codon, or has the corresponding anticodon that's going to match the codon in the sequence. So there is a tRNA for selenocysteine that matches the UGA sequence, but that tRNA, it actually first gets loaded, it gets charged with serine. And so it gets loaded with serine, and then the serine gets modified. It gets modified, it gets that selenium part added on. And so this is another distinction why we don't typically classify it as one of the like normal traditional amino acids, but it does exist in our bodies. What doesn't exist in our bodies, however, is this pyrrolysine, which is found in bacteria, some bacteria in archaea, and it is actually made out of lysines. It's pretty cool stuff. It actually does have its own, it does get charged with this pyrrolysine and that sort of thing, but we don't have it in our bodies, and it's only found in a few species that we know of. But getting back to the idea of the structure of these amino acids themselves, we're in our bodies, our amino acids, we use the L forms, and this refers to stereochemistry, which refers to like what direction things are sticking out in space. So basically, like if we say something is chiral, it has like non-superimposable mirror images. So kind of like your right hand and your left hand, they're non-superimposable, but they're mirror images. And so molecules are like that too. And in the case of our amino acids, we have the L form. You'll see D form in a couple of weird cases, such as in bacterial walls, which is pretty cool. But anyway, in some like therapeutics and stuff, they're looking at using like the other like reverse type of things so that they're more resistant to degradation. There's some cool stuff being done in the chirality field, so keep an eye on it. But in our bodies, we're using this L form, and so all of our cellular machinery is basically like, it would be like a left-handed baseball mitt or something like that, where our machinery is specialized to use that one form. Okay, so those are the basics of amino acids. We start with those 20 basic subunits, and then those a couple exceptions. But then there also can be post-translational modifications. So these are changes that happen after translation, which is the process of putting together these amino acids, linking them together to form proteins. So when you get modifications that get made after they've been linked together, we call those post-translational modifications. And some common ones are like methylation, acetylation, phosphorylation. You can have oxidation and hydroxylation. So, but one of the key things is that which of these modifications you have is going to be limited by which amino acids you have. And so certain amino acids have a greater tendency to be able to be modified in these ways. So we can talk about these. We can talk about nucleophilic amino acids. So too technical to get into here, but basically these amino acids, so you have serine, threonine, tyrosine, cysteine, lysine, adenine. These ones have more reactive groups that can then get modified in various ways, and more on that in other posts. But going back to where we were, let's talk about how these amino acids actually get linked together. Sorry. So first off, what are these amino acids made up of? These amino acids, like all molecules, are made up of atoms, which are the basic building blocks of elements. So carbon, hydrogen, nitrogen, oxygen, sulfur, phosphorus, all of these various elements, they come in like units of atoms. And inside of these atoms are even smaller parts called subatomic particles. And so you have this dense central nucleus, which contains these positively charged protons and these neutral neutrons. And then surrounding them, you have this electron cloud. So you have this cloud of these negatively charged subatomic particles called electrons that are kind of like whizzing around. Atoms can share pairs of electrons in order to form bonds. So they share a pair for a single bond, 2 pairs for a double bond. And sometimes they don't share fairly. And in this case, we can get uneven distribution of charge, which we call polarity. So because those protons are the positively charged protons are stuck in place, but the negatively charged electrons are in this like cloud. This means that when these electrons are shared, they can kind of get swayed towards the nucleus, so towards the positively charged part of one of the atoms that is sharing them. This is a great example of this is water. So you have the oxygen, which is what we call electronegative. Something that's electronegative is going to be hogging the shared electrons. So the oxygen is hogging the shared electrons, making it partly negative, making the hydrogens partly positive. So you have this polar molecule where you have this separation of these partial charges. When you have separation of partial charges, when you have full charges, then you get the possibility to have intermolecular attractions. And so these attractions play a huge role in what type of interactions that molecules make, as well as where molecules want to hang out. So we'll see this again and again and again, that either with full charges, with partial charges, even with temporary charges. So basically the electron, one molecule moves close to something and then that kind of like gets them to all like shift their electron clouds because those electrons are really just whizzing around. We can never even predict where they'll actually be. They're just places where they're more likely to hang out. And so they're more likely to hang out by something positive and away from something negative. And so therefore, like these clouds can get kind of like shifted and this allows the molecules to sync up and you get this like induced dipoles and various things. So basically there are a lot of ways that molecules can interact and what ways that they have to interact is going to depend a large way in what their side chains are made up of in the case of the amino acids. One of the key things that you'll see with amino acids where this comes into play is this idea of the hydrophobic effect. So as I mentioned, water has this high polarity, and this means that because water has these partial positive and partial negative parts, it's going to be attracted to itself because it has these parts will be attracted to the oxygens of other waters. But the oxygens of other waters will be attracted to these, and so you can get these water networks. Water will let in things in its network if those things also have something to offer. So if those things have some sort of positive charge or they have some sort of negative charge, even if that charge can be a full charge as you see with an ion, or it can be a partial charge as in the case with the water where you have like a polar molecule. We call things that water likes hydrophilic, and these things water will like let into its network and be soluble. We also have things that water doesn't want to hang out with. So things that water doesn't want to hang out with involve when the molecules are like sharing fairly. So if you have like a hydrocarbons, hydrogen and carbon, they share pretty fairly. And so these groups tend to be like non-polar. Whereas if you have you can add functional groups, which are basically things other than hydrocarbons, and these often are polar. And this allows you to add polarity to a hydrocarbon-based molecule. So when you see amino acids that have just like hydrocarbon chains, those are going to be hydrophobic. So water's not going to want to hang out with them. And when you see ones that have like charged groups or polar groups, now these are going to be hydrophilic. So water will hang out with them. This has huge, huge, huge consequences when it comes to how proteins fold, because when proteins are folding, they're going to fold so that the hydrophilic parts are on the outside and the hydrophobic parts are in the inside. And this is going to be a huge driving force in protein folding, is the water kind of like excluding those hydrophobic amino acids. So things like isoleucine, phenylalanine, leucine, valine, methionine, tryptophan, these are very hydrophobic amino acids. And so when the protein is folding, the water is going to exclude them and kind of like pinch around them. They're going to get grouped together in the center of protein, minimizing their contact with the water. If you end up putting one of these on the outside of a protein, you can have dramatic consequences, and this can be a cause of disease. So a key example is the hemoglobin protein. So this is a protein that carries oxygen throughout our blood. In sickle cell anemia, there's a mutation on the surface of this protein from a glutamate, which is negatively charged, to a valine, which is nonpolar. This then makes this like hydrophobic pocket on the surface of the protein that causes it to link up and which can clog blood vessels, leading to basal obstruction. So basically the blood can't get through the vessels, and then you can have organ damage, great pain crises, et cetera, et cetera. So not good. So getting back to the amino acids and how they link together. As I mentioned, they link together through what we call peptide bonds using the amino group and the carboxylate group. And then on each end, one end has a free N terminus, so which has an amino. One end is the N terminus, which has a free amino group. So we call it the N terminus because amino, it has a nitrogen. And on the other end, we have a carboxyl group, which we call the C terminus because it has that carboxyl group. So typically you're going to see this amino group be protonated and thus positively charged, and the carboxyl group be deprotonated and thus negatively charged. So we link these up through these peptide bonds. If you link a few of them, you get an oligopeptide, so oligo meaning few. If you link a lot, you get a polypeptide, so poly meaning many. And when you fold that up, you get a functional protein. And the way that it folds up is going to be influenced by the properties of those amino acids. The linkages between these amino acids, these peptide bonds, they have this kind of cool property in that they have this resonance. So they have the electrons are actually going to be shared throughout among these molecules rather than just between two atoms. So I said like atoms can share a pair of electrons to form a bond, and two pairs of electrons to form a double bond. Sometimes they can actually share those electrons with more than one atom. When this happens, you get this thing called conjugation or resonance, where you can draw it as having like a double bond here or a double bond here, but really you have this partial double bond character throughout. It's really this like communal sharing. Why this is consequential is because you can't rotate around a double bond. And so you also can't rotate around this partial double bond. And so this part is like stuck in place and you can only rotate around it. And so you end up with this like chain of planes. When you have this chain of planes where you have this rotation that's limited, the rotation is going to be limited even further by the nature of this side grain, by the nature of this R group. So if you have something that is very small, say, you'll have more flexibility. And if you have something that's bigger, you're going to have less flexibility. But that's not the only determining factors. It also matters where the bulk is located. So if you take for example, let's see, what do we want to take for an example? Okay, we want to take leucine and isoleucine. these are the same size in terms of like, if you would look at the molecular weight, but with isoleucine, you have the branch is closer to the backbone. So basically you have more bulk, you have your bulk shifted closer to the carbon backbone. This is going to hinder the rotation more than if you had it further out. Speaking of leucine and isoleucine, so these are two examples, and then there's also valine of branched chains amino acid because they have a branch in a hydrocarbon chain. And these actually go through a lot of the same processing steps, and mutations in the processing machinery for these can lead to diseases such as maple syrup urine disease, and more on that in my other posts. There are also a couple of weirdos when it comes to the backbone angles. So if we look, glycine, you might think, okay, well, it is small, you have the most flexibility. It should be able to be like just the most generic, right? Actually, we typically use alanine as the most generic because glycine, it turned out, is so flexible, it can take on all of these. It can take on a lot of weird angles. Contrast this, however, with proline, which is the most restricted because it has its backbone kind of like twisting back onto itself and hooking up with its backbone like that. Okay, so these angles that the protein can take matter, that the backbone can take matter because they have to take distinct like backbone angles when they form their key secondary structure. So when we talk about layers of protein structure, we can talk about the primary structure, which is just a sequence of amino acids. Then they fold up, you get the secondary structure. This involves interactions between the backbones. Then these then the side chains get involved and you have further interactions. You get these tertiary structure, which is going to be a single chain of a protein that's folded up through interactions involving both the backbone as well as the side chains. And then if you have multiple chains in your protein, such as, for example, when we looked at hemoglobin, it had these multiple chains in this to form the whole protein. And so this is what we call a quaternary structure, where it has multiple chains involved. And so then your structure is going to involve interactions between those chains, as well as the interactions within those chains. So you have these multiple layers of protein structure. The backbone, so that's where you get the secondary structure. This is coming from interactions between the backbone, more specifically these like hydrogen bond interactions, which happen basically. A hydrogen bond is just a special case of a partial charge attraction, where you have a hydrogen that's attached to something electronegative, so it has something that's hogging electrons away from it. So hydrogen is going to be partly positive, and then the oxygen is going to be partly negative. Or it could also be like a nitrogen or things like that. So in the case of this peptide backbone, it would be a nitrogen pulling these electrons, making this partially positive. Then this would be your donor, and then you have an H bond acceptor, which is something electronegative with a lone pair of electrons. So this can be an oxygen or nitrogen with a lone pair. In this case, it's an oxygen with a lone pair. This allows you, because you have this regular first pattern basically of these donors and acceptors for these bonds, you can get these characteristic secondary structures involving alpha helices and beta sheets. So beta sheets are made up of beta strands that come together. And basically, the key thing is that these have distinct backbone angles that get taken. So those angles between those planes in the backbone, and different amino acids have different tendencies, different abilities to take on those angles more easily. And so this means that you'll have some amino acids will tend to form helices more, some will tend to form beta strands more, et cetera, et cetera, et cetera. So how else can we characterize amino acids? So we've talked about how we could talk about polar and unpolar, or polar and non-polar, and how the non-polar ones tend to be a little hydrophobic. And so they'll tend to be found in the inside of a protein, and then the polar ones tend to be hydrophilic and on the outside of a protein. We can also talk about them. The polar ones also include, or the hydrophilic ones also include, the acidic and the basic, which are the charged ones. So let's get a little into the terminology of how we consider something like acidic or basic. So acidity is a measure of proton concentration. So we talk about the pH of a solution. The more protons there are available free floating, the more acidic the solution, and the lower the pH because it's an inverse log scale. So the more protons you have, the fuller your like proton jar, I like to think of it, the lower your pH. And conversely, the fewer protons you have, the higher the pH, the more basic or alkaline the solution is. This matters because when there's more protons around, molecules that have a protonatable group, so molecules that can take a proton, then will take a proton if there's one around. Whereas if there's not many protons around, then they won't. What their tendency to do this also depends on how much they want the proton, which comes from a term called the pKa. So that's a measure of like the acid strength. So an acid is something, in one sense, that an acid is something that will donate a proton, and a base is something that will accept a proton. When we talk about the acidic amino acids, we're talking about aspartic acid and glutamic acid. But typically, because these have a really low pKa, so the pKa is the value at which half of the molecules are negatively, or half of the molecules are protonated, half are deprotonated, or if you were to look at a single molecule, would have a 50-50 chance of being deprotonated. So these molecules these molecules like go back and forth between this aspartic acid form and this aspartate form. Similarly, how we talked about the carboxylic acid group of amino acids can go back and forth between the carboxylic acid and the carboxylic group, but you'll typically find it at the carboxylic group. Because it has a low pKa, it'll tend to be deprotonated. Because you're going to be above, if your physiological pH is about like 7.4 or so, you're going to be way above the pKa, which is about 2. So remember, the pKa is the pH at which half of it is going to be deprotonated. And if you go to a place where there's fewer protons available, so if you go higher in the pH, which is 7.4, is much higher than 2, then we're going to be deprotonated. And in fact, we're going to be way deprotonated, because each pH unit represents a 10-fold difference in the amount of protons. And so you're going to have in your body, this is going to be deprotonated. And the same holds true when it comes to aspartic acid and glutamic acid. So we're almost always going to find them in their aspartate and their glutamate forms, which are the conjugate base forms of them. Conversely, we have, so although we call them like acidic amino acids, really it's that they've already acted as acids and now they're in their conjugate base form. So don't get confused about that. When we talk about the like quote unquote basic amino acids, we're talking about histidine, lysine, and arginine. So lysine and arginine, you can see that they have these very high pKa's. And so the pKa is much higher than the pH that our cells are typically at. As a result, they're going to be protonated almost always. And this is going to make them positively charged. With histidine, you can see that you're pretty close to the pKa is pretty close to the pH of our bodies. And so you're going to have histidine. You're going to have, although predominantly the histidine is going to be in the deprotonated state, you're still going to have a significant portion of it be protonated. And this can, but histidine can go back and forth, and this can allow it to play important roles in the body, especially like in enzymes and really cool things. So these are sometimes charged, but they can go back and forth and they can act. This is, although I'm talking about like the pH of your body or whatever, but you actually inside of enzymes, inside of molecules, inside compartments, there can be differences in the pH. There could be local differences in the pH. There could be, say, something that's next door that has a negative charge that's kind of like pulling on it. And as a result, you can alter this pKa, you can also alter the local pH, and therefore you can have these molecules give and take protons more readily, and therefore serve as important roles in like giving and taking protons in reactions. And so a lot of protein active sites, like enzyme active sites, so enzymes are molecules that like speed up or catalyze reactions, and you'll see these positive or negative charge residues in there often, as well as those like nucleophilic residues we talked about. Another way that we can characterize amino acids is by like what the aromatic amino acids. So these are the ones that have these aromatic rings in their side chains. So we talked about resonance before, like electron conjugation, which we talked about this in the context of the peptide bond, where you have the sharing of electrons between multiple, more than two atoms. This can also happen in between more than three. So it can actually happen between a lot of them. And if it happens in a ring, we call this molecule aromatic. There are three aromatic amino acids. So you have tryptophan, phenylalanine, and tyrosine. And these have these aromatic rings. And these can have important functions because they allow them to form different types of interactions through like pi bonds, which are these where you have this like delocalized electrons. They can do various things, interactions with say like DNA and RNAs, aromatic rings, various really cool stuff. They also have this really helpful property that they absorb UV light well, so especially tryptophan. These rings, when you have this resonance, it has a consequence of like lowering the amount of energy it takes to excite an electron. So more on this in my post on like fluorescence and UV and various things. But what's important here is that it'll absorb UV light, so ultraviolet light, so really energetic light that we can't normally see. It'll absorb it, and we can detect the absorption using a machine that basically measures how much of the light is getting through. And we can convert that to the protein concentration, because the higher the concentration of your protein, therefore the more tryptophan. So we'll be assuming your protein has some tryptophans, and therefore the higher the absorbance. And because different proteins have different concentrations of these aromatic amino acids, you can calculate this thing called the absorption coefficient or extinction coefficient for your specific protein and the sequence of amino acids it has. And you can use that to then calculate the concentration of your protein based on its absorbance. We can characterize amino acids based on their like more functional things like in the body, as well as more kind of network thinking. So we can talk about like what can we use amino acids for other than making proteins. So we can break down these amino acids and we can alter them into different forms that can be used for different things. When it comes to our metabolism, so like the making and breaking of molecules, we often think about in terms of pathways, but really things are more like networks. So if we can't make something directly from something, that doesn't mean we can't like use something from it to make something from it. And often when we talk about like whether it can be used for something, we are talking about whether it can be used for something in like an energetically favorable way, if that makes sense. Basically, if you get a net output of the molecule versus, yes, you can convert it into that molecule, but you end up using up other molecules and there are various complications when it comes to whether we determine whether we call something, say, glucogenic to be used for making sugar or ketogenic to be used for making lipids like fatty acids. And so I have more in this in my posts on those. But it's not that these things can't be used to make the other. It's like somehow these parts of molecules can usually always be used to make one thing or another. It's just that if you're using up in the process, say you have to add something else, then it wouldn't be considered. So though I'm not making any sense, but just know that it is complicated how we define these terms, and not everyone always agrees on these terms. But so you know, this is how it's typically classified, so that you have the glucogenic amino acids, which make something that go into this tricarboxylic acid cycle. And then you have the ketogenic amino acids, which can make these ketone bodies and be used to make lipids. But that doesn't mean that you can't make lipids out of glucogenic amino acids, et cetera, et cetera. And it also depends on what enzymes are available. It's complicated, and I'm not going to get into it. But if you're confused, I apologize, but it is confusing. Okay, so don't feel bad. Then finally, we can talk about how we get these amino acids. We can classify them in terms of essential or non-essential. And so these, we need them all. So they're all like essential in that sense. But when we talk about dietary essentiality, we're basically saying, do we need to get it from our diet or can we make it in our own bodies? And so if something is non-essential, our bodies can make it from other things. And if they're essential, then we have to get it from our diet. And so, yeah, so those are the basic ways we can classify amino acids, and I hope that this helped you understand them.